Hey guys, welcome back to Now I Know. Today we are talking about population genetics. In that we are talking about Hardy Weinberg equilibrium or Hardy Weinberg principle. Now this is one topic where I've seen students getting confused in like small points. And this is one of the topic which actually assures you marks in exam. The population genetics problems based on Hardy Weinberg equilibrium. It's really easy once we understand, and trust me, it's very interesting. So today what we will do in this video is first we will try and understand what this Hardy Weinberg equation is, how it helps to solve the genetic population problems, and in the end of a video we'll take couple of sample problems so we are clear how to apply this and solve the population genetics problems. So let's begin. Alright, so the first thing is we are talking about population genetics, right? So I think it's important that we first understand what is population genetics. So population genetics is study of genetic structure of a population. Alright, what does this mean? Let's break it down and make it easy. It says it's a study of genetic structure of a population. So what is a population first of all? How do I define population? Population is nothing but a group of individuals of the same species that can interbreed. Simple to understand. Then what is this genetic structure? What we are studying in this population? Now genetic structure means it is going to tell you about the genetics of that particular population, and if we want to study genetics or genetic structure of the population, it will be let's break it down into two parts. Study of alleles, right? Because we know that all the genes would have different variation, right? Alleles would be there in a population. So genetic structure comprises of alleles. What is the frequency of particular allele? If I'm studying allele X, what is the frequency of this allele in any particular population? How much of that allele is distributed in the population? And when you talk about alleles, the combination of alleles gives us genotype of an individual. Simple, right? Based on that we have homozygous dominant genotype, homozygous recessive genotype, and heterozygous genotype, right? Three types of genotypes we can have. So what is the frequency of each type of genotype in the population? So genetic structure is nothing but the frequency of alleles and genotypes present in any particular population. Now why we need to do that? Imagine you are studying a disease, okay? Let's say you are studying a recessive disease. So you know that you need two recessive allele for that particular disease to appear, and if a person has one of the recessive allele, that person would be a carrier for that particular disease in a population. So by this population genetics, we can study what percentage, what is the frequency of this allele or the affected individual, or what is the frequency of carrier in that particular population. Basically what we are going to do, what we are going to talk about in this video throughout is only about alleles and genotypes. Okay, so I think we are clear about this. If you want to know a little more about alleles or heterozygous, homozygous, what is the difference, I'll put the links on the screen. We have talked about it. Now this allele and genotype frequency, is it constant in a population? No, right? It changes over the time. It is not possible that the allele frequency and genotype frequency in any given population can be stable naturally, because we have one or the other evolutionary force acting on it. I'm just going to list the factors over here that actually affects the frequency of alleles and genotypes naturally. We are not going to talk about it in detail. If you guys want some other time, we'll talk about it. But let's just list out what all factors can be there. Let's say for example mutation, migration, natural selection, genetic drift, and non-random mating. Basically, these are the factors that actually affects the frequency of allele and genotype. Like, let's just say, for example, if there is mutation, there is a spontaneous change in DNA that is going to affect your alleles, that is going to affect the genotype of a population. So, any factors, at least one of these factors would be affecting a population at a time. So, generally, naturally, alleles and genotype frequency keeps changing over the time. Okay, that is one factor that we need to understand. Why? Because we are, constantly under the influence of these evolutionary factors. So, it is difficult when you want to calculate the frequency of alleles and genotypes for such population, right? So, what we can assume, let's say for example, if these factors are constant, okay, or there is no change due to this evolutionary factors, none of these factors are working on a population, then that population has a constant or the same frequency of allele and genotype, right, over the period of time. It wouldn't change because we are assuming that none of these factors are working on a population, so there will be no change in the allele and thus there will be no change in the genotype. That's what Hardy Weinberg equilibrium states. It says that frequency of alleles and genotypes in a population remains constant over time in the absence of any evolutionary influences. So what this Hardy Weinberg principle says is that frequency of alleles and genotype in any population will remain constant over the time if there is no evolutionary influence working on it. None of these factors, if they are working on a population, it will not affect the alleles and thus it will not affect the genotype. Easy to understand, because if any of these forces, even any one of these is available or working on a population, is going to change the frequency of genotype. Now this looks very ideal condition, right? Is it possible naturally? Is it possible that naturally none of these factors would be working on a population? No, right? In a real world it's not possible. So then why are we using this Hardy Weinberg principle when in the real world it is not possible at all? As I said in the beginning, right, if you make assumption for that particular population that none of the factors are working on a population, we can actually calculate the frequency of particular alleles or genotype in a population, which is very helpful to understand if you're working with some disease. You're working to find out the frequency of particular allele, how many carrier of that disease is present. This is going to help us. So we are going to assume this. We are going to make assumptions like there is no mutation in a population, there is no migration in a population, no natural selection, no genetic drift, and there is a random mating in a population. Okay, now I'll tell you the first point and very important point that you need to remember throughout the Hardy Weinberg equilibrium, whenever you are dealing with Hardy Weinberg equilibrium, is it talks about frequencies always. Alright, we are talking about alleles and genotypes, right? So it always talks about frequency of alleles and frequency of genotypes. Right, not the numbers as it is, but the frequency of allele and frequency of genotype. Okay, this point is very important throughout. Whenever you are dealing with Hardy Weinberg equation, you need to remember, talk about frequencies. Okay, numbers in frequencies, frequency of allele and frequency of genotype. Okay, that's one point that you need to remember always. Now let's start with the alleles. Right, when we talk about allele, we know that we have two types of alleles. One would be dominant and one would be recessive. So the dominant allele, in case of Hardy Weinberg, we denote it as small p. Alright, and the recessive allele, we will designate it with small q. As I said, Hardy Weinberg equation always talks about frequencies. Right, so that means frequency of dominant allele is denoted by small p, and frequency of recessive allele is denoted by small q. Okay, either there is p or there is q that is present. Okay, there cannot be any more allele as of now. There can be more, but as of now we are assuming that there is only p and only q is present in a population. So if there is only p and q is present in the population, that means together they make 100% of a population. Simple enough to understand, right? What I mean is, if I have let's say for example I have 70%, that means 70 people in a population who are carrying dominant allele p, then rest 30 would be q, of course, because as I said there's only p or q present in the population, so it has to be one or the other. So that means p plus q makes my 100% of population. So always p plus q equals to 1. Very, very important. Number 1 to remember. As I said, either p or q is present, that means together they make complete population. So p plus q, the sum of frequency of p and q should equal to 1 always. Equation number 1 that you need to remember. Frequency of dominant allele plus frequency of recessive allele equals to 1. So that means if I know frequency of any one allele, either the dominant allele or the recessive allele, I can find out the frequency of the other allele, right? Because simply what I need to do is, say for example, I know frequency of recessive allele q, maybe that is 0.3. Okay, so what I need to do is p equals to 1 minus q, that means 1 minus 0.3, so p equals to 0.7, right? And if I know the same way, if I knew what is frequency of dominant allele, I can find out the value of q. So this equation is going to help us a lot. If we know the frequency of one of the allele, we can find out frequency of the other allele, and we are going to use it for that purpose everywhere. So remember, p plus q equals to 1. Now, if there are only two alleles, there are only two combinations present in a population, p and q. This is how I tell my students, because I think this makes it very easy for them to understand and remember the actual equation. So there are only two alleles present in a population, p and q. So when you cross them, p q allele cross with p and q allele. So when p and q allele is crossed with p and q allele, what all combinations we get? You know, make simple Punnett square. You will get p into p, p into q, p into q, q into q. Look at this, p into p, right? That means p square. What is p? P is frequency of dominant allele, right? Frequency of dominant allele. So we have two p's. That means we are talking about two dominant allele together. And what do we call two dominant allele together? Homozygous dominant, right? When there are two dominant alleles together, that condition is homozygous dominant, right? So p square, as I said, it always talks about frequency. So p square gives us the frequency of homozygous dominant. Alright, now look at this. What is this? Q into q, that is q square. And what is q? Q is frequency of recessive allele. So there are two q's, that means there are two recessive allele. And what does two recessive allele makes together? It makes homozygous recessive. We are talking about frequency, so frequency of homozygous recessive. Q square equals to frequency of homozygous recessive. So now what are we remain with? PQ, PQ. That means 2PQ. P is for dominant allele and Q is for recessive allele. So when you have one dominant allele and one recessive allele, what is that condition? It is heterozygous. As I said, we are talking about frequency. So 2PQ gives us the frequency of heterozygous, right? What are these? These are nothing but the genotypes. So we started with alleles, right? Allelic frequencies P and Q. What are the genotype frequencies then? P square is the frequency of homozygous dominant, Q square is the frequency of homozygous recessive, and 2PQ is frequency of heterozygous. Now, as I said before, population has only P and Q, so sum of it should be 1. Similarly, sum of all these genotypes also should be 1. So that means P square plus 2PQ plus Q square. All of these genotypes should equal to 1. Remember the second formula, very, very important. Sum of all the genotypes should be 1. So P plus Q equal to 1 is useful when you want to find out the frequency of alleles, because P and Q is frequency of allele. But when you want to find out the frequency of genotype, you are going to use this particular formula. P square plus 2PQ plus Q square equals to 1. Where P square is frequency of homozygous dominant, Q square gives me frequency of homozygous recessive, and 2PQ gives me frequency of heterozygous. Alright, so once I know what is my P and Q, what is the frequency of allele, I can put this value of P and Q in this equation and can find out the frequency of genotype, right? So this is all you need to remember. In Hardy Weinberg principle, when you are solving population genetics, you need to find out two things. Okay, one is the allelic frequency and the second one is genotype frequency. For allelic frequency, you will use P plus Q equal to 1 formula, and for genotype frequency, we are going to use P square plus 2PQ plus Q square equals to 1. Now, let's keep this in mind and try to solve some sample problems. Alright, so let's start with a simple problem. Before we start with this, two tips I want to give you. Number one is, whenever you're looking at population genetics problem, whenever we have to apply the Hardy Weinberg principle, we are always trying to find out the allele frequency and genotype frequencies, right? And we saw both the formulas. So keep in mind that you need to find out, make a list of it, okay, this way. You need to find out the allele frequencies, and that is nothing but when it is dominant allele, frequency of dominant allele, you need to find out P. And it is recessive allele, frequency of recessive allele, that means you need to find out Q. Then you need to find out the genotype frequencies. And genotype frequencies we just saw, that means you need to find out P square, that gives me frequency of homozygous dominant. We need to find out 2PQ, that is frequency of heterozygous condition, and we need to find out Q square, that is the frequency of homozygous recessive. Okay, so these 5 things we will look at generally when we are dealing with such problem. And second tip that I want to give you is, always look at the homozygous recessive individuals in a population. Okay, always locate homozygous recessive individual first. Either they will give you the homozygous recessive individuals, that means that is the value of Q square for you, right? Why? Because homozygous recessive condition is only possible when you have 2 recessive allele, Q into Q. That means the number of individuals who are homozygous recessive in a problem is the number of Q square. Okay, not Q. Don't get confused over here. Okay, this is one point where students get confused. Q is only one allele. When you have individual, when you're talking about individual with homozygous recessive condition, that individual has 2Q allele. That is why that person is showing that homozygous phenotype. So the individual who are having homozygous recessive condition is the number of Q square. Alright, so now let's look at the problem. It says that in a population of 100 cats, total population is 100, 84 cats are black and 16 cats are brown. Brown is a recessive phenotype. Okay, brown is a recessive trait. Calculate the allele frequency and genotype frequency of this population. So here, by looking at it, I said always locate Q square first. So this is the number of q square, 16, right? Because they are showing the recessive traits. So q square over here is 16, right? Now I just told you in the beginning, whenever you're dealing with such problem, Hardy Weinberg always talks about frequencies of alleles and genotypes. So this is simply a number of individual. What you need to do is you need to convert this or take frequency of this. And how you can do it? Just simply divide it by total number of the population. So I have q square. I need frequency of q square. Now what I can do is 16 by 100 is total population equals to 0.16. So frequency of q square is 0.16. This makes half of my work easy. I know q square, frequency of q square. Simply I can find out frequency of q from this. Just take square root of this value. So it is 0.4. Frequency of q is 0.4. Now do you remember, if I know frequency of one allele, I can find out frequency of another allele from this equation, p plus q equal to 1. So I can find out frequency of p equals to 1 minus 0.4. So p equals to 0.6. So frequency of p is 0.6, right? So I got my allele frequency. Frequency of p is 0.6. Frequency of q is 0.4. And this is true. If you just add them up, of course, you are going to get sum of it as 1. Now what we are left with, it has asked for genotype frequency. So we need to now find out these three values. Since we know p and q, this is going to be very easy. So let's find out p square. P square would be 0.36. Q square will be 0.16. That is what we got initially. And 2pq would be 2 into 0.4 into 0.6, that equals to 0.48. And when you take sum of all these three genotypes, you will get 1. So what this says is, in this population we said 84 cats are black, right? 16 cats are brown, that is homozygous recessive. We got it. 84 cats are black. Out of this 84 cats, 0.36 means if you take individual, that becomes 36, right? 36 cats are homozygous dominant and 48 cats are heterozygous for that particular trait. Now let's take a little different problem than this. So it says that cystic fibrosis is an inherited recessive genetic disorder. Look at it. It says it's a recessive genetic disorder which is occurring in 1 in 1700 newborns. What is the frequency of carrier of this disease in the population? Now if you look at this problem, it is just asking us to find out the frequency of carrier of that particular disease, right? And since they are talking about recessive genetic disorder, that means carrier would be a heterozygous condition where it contains one recessive allele. So it is actually asking us to find out 2pq value only, right? So to find out 2pq, that is, it is asking us to find out only one genotype. But in any case, we will have to find out the frequency of alleles, then only we can find out this genotype. So as I said, just make a list of the things. Allele frequencies, so genotype frequencies, we will have to find out p square, 2pq and q square. They have asked us carrier for that particular disease, so we will talk about only, like, we need to give answer for only 2pq. Alright, so can you locate q square over here? As I said in the previous example, always try to locate q square. So this says this recessive genetic disorder occurs 1 into 1700 newborns. Since we are directly taking 1 by 1700, this becomes my frequency, right? Isn't it? Directly, if it was given only one individual, if I say it is my q square. But now when I am taking frequency, I will divide it by 1700, the total population. So they are saying it is 1 into 1700, that gives me frequency of q square. This is my q square, right? So 1 by 1700 is 0.0006. Alright, so if I want a frequency of Q, simply take the square root 0.024. So I got frequency of Q. I can now easily find frequency of P. So frequency of P should be 1 minus 0.024. So that equals to 0.976. So when you add these two, it will become 1. So now I have got my P and Q allele frequencies are done. Easily I can find out 2PQ, that is the carrier for the disease. So 2PQ should be 0.047. So this is the number of, now they have asked for frequency of carrier. Okay, so this is the frequency, 2PQ is frequency. In case they had asked me number of individuals, what I need to do is I need to multiply this number with the total population that they are talking about. Here it is 1700. So the number of individuals that are carrier in this particular population would be 0.047 into 1700. That becomes 80 individuals in the population are carrier. Okay, and in case you had to find out other frequencies, you simply need to take square of P, frequency P, that will give you P square. For Q square we have already found out, so this is the Q square. So that's how we solve the population genetics problem using Hardy Weinberg equilibrium. All you need to remember is allelic frequency P and Q, genotype frequency is P square plus 2PQ plus Q square. Always try to locate the Q square. If individuals are not given and they have talked about only single recessive allele Q, try to locate that. That will make the problem very easy to solve. So that's all for now. I hope this video was helpful. Do subscribe to the channel for new video every week, and I will see you next time. Until then, keep learning.